take a look at meiotic metaphase 1. Now, after diakinesis of meiotic prophase 1, the chromosomes have been pushed to the metaphase plate. They've been aligned on the metaphase plate. So I have the metaphase plate diagrammed here. Remember, the metaphase plate is an imaginary line down the center of the nucleus. So, and here we can see that we have both tetrads aligned on the metaphase plate. And unlike mitosis, the centromeres are found on either side, right, of the metaphase plate. So if we see centromeres side by side, one on either side of the metaphase plate, we know we are not talking about mitosis. We know we are talking about metaphase 1 of meiosis, or meiotic metaphase 1. So at this point, you should ask yourself, what if I asked you to diagram, these chromosomes during mitotic metaphase? What would that diagram look like? So essentially, we don't know what order the chromosomes will be in along the metaphase plate, but they will be one on top of the other. So no tetrads. Tetrads are only found during meiosis. So you would diagram something like that, with the centromeres right on the metaphase plate. And you could add the microtubules if you wanted to. So another point I want to make here, because this will be helpful for later in the course, is that, okay, so there are 4 chromosomes here, right? So quick diagram of each of these. I want to name these because it should help us see something. So I'm going to name these guys chromosome 1, these chromosome 2. Let's say this is from the paternal parent, this is from maternal, this is from paternal, this is from maternal. Now, in a previous video, we talked about how meiosis needs to reduce the number of sets of chromosomes. Not necessarily the number of chromosomes. It does that, but it needs to reduce the number of sets, and it needs to produce meiotic products with complete sets. And the way it does that is by pairing up the homologous chromosomes, like here and here. And then it takes one member of each pair and segregates them from each other. So we have, let's say, the pair at the top, this chromosome on the left, it's going to go towards the left, the other one's going to go towards the right. Same thing for the bottom. The one on the left is going to go to the left. The one on the right is going to go to the right. So it's able to, by doing this, by pairing them up, and making one go in one direction and one go in the other direction, it can make sure it forms complete sets of chromosomes. So another thing I wanted to point out is that we don't really know if this one over here, let's say, let me clean up this diagram a little bit. We don't know if this one right here is, say, 1P, and then this one is 1M, or maybe this one's 1M and this one is 1P. We don't know that. So if this one is 1P and this is 2P and this is 1M and this is 2M, what that means is 1P and 2P are going to go together into one meiotic product, and 1M and 2M are going to go together into another meiotic product. However, something different could happen, right? So what else could happen? So let's say 1P up here might be found on the same side as 2M. And that would put 1m on the same side as 2p. In this situation, 1m and 2p would go in one direction, and 1p and 2m would go in the other direction. So the way the tetrads align and the orientation they adopt on the metaphase plate relative to one another is completely random. So whether 1p is found on the same side as 2p or 2m is completely random. So we'll call that random orientations or random arrangements of the tetrads on the metaphase plate occur. So that process is completely random, and that leads to independent assortment. So we will talk about independent assortment next week. And that's what that's what causes independent assortment. The fact that the way the tetrads align on the metaphase plate relative to one another causes independent assortment. Okay, so I think that's it for meiotic metaphase. Essentially, the tetrads are aligned on the metaphase plate.